koto, no me hare mai. Thanks for joining tonight. I'm Jodie Clark from the Alumni Relations team at the University of Auckland, and I'm delighted to welcome you to another great talk in our Raising the Bar Home Edition series. Over the past 4 weeks, leading academics from the university have shared their research and discoveries on a wide range of subjects, from democracy to wine to genetics. Tonight is our penultimate talk of the series, and we'll hear from Professor Justin O'Sullivan, Deputy Director of the Liggins Institute. Justin's talk will shed light on how we can use DNA information to not only improve our well-being, but also extend our lives. Joining Justin as our MC is Professor Wayne Cutfield, a previous Raising the Bar speaker and colleague of Justin's at the Liggins Institute. Wayne will introduce Justin in more detail and then lead the Q&A session with questions submitted by you, the audience. If you've missed any of our previous talks, you'll find recordings of each on our alumni website. Next week, our final talk explains what word of mouth is and how you can use it. Registrations are still open for the talk. Now, though, it's time to sit back, relax, and find out how we can all extend our lives. Over to you, Wayne.
Kia ora tātou, and thank you very much, Jodie, for that warm introduction. Just a minor housekeeping matter before we get underway. Can you please use the Q&A button for your questions? Don't put your questions into chat. We won't see them there. And if you see a question that you particularly like, you can upvote it with a little heart image, and that will get the question asked a bit quicker. I'll address the questions to Justin at the end of his presentation. There are 7.9 billion humans on this planet right now. 7.9 billion. 99.9% of their DNA is identical. 99.9%. Just 0.1% difference in DNA determines the differences and makes us unique. But in that 0.1% is also that where we share our risks of diseases that we will develop. It gives me great pleasure to introduce a very good colleague of mine, Professor Justin O'Sullivan, the Deputy Director of the Liggins Institute and one of the country's leading molecular microbiologists and research molecular geneticists. Justin is going to talk to us about precision medicine, about the way in which our genes determine our risks of diseases and how our genes might determine how we might respond to drugs in the future, and how our genes may determine our longevity. His topic tonight, the untapped potential of DNA to personalize your healthcare and prolong your life. Justin, the platform is yours.
Thanks, Wayne. Right. Pina koutou katoa. Ko Justin takuingoa, ko Sullivan takuinga whāwanau. Ko o te manatuaki rua putahi o Liggins I Waipapa Maka Road. Ko taku ko ngā mahi mātai iranga. Ko taiatia aho. Welcome, everybody. I'm Justin O'Sullivan. I'm the Deputy Director at the Liggins Institute at the University of Auckland. And I'm a scientist who's interested in genetic variation. And tonight, I'm a little nervous. But I'm going to talk to you tonight about genetics, about precision medicine, as Wayne said. So as we start our talk tonight, what I would like you to do is just take a look around you. What do you see? When I look around myself, I'm in my office at the Liggins at the University of Auckland. I see things here that look permanent. I see a computer screen I'm looking at. There's my desk, there's the walls. They all look permanent. But that permanence is an illusion. If you look again, it doesn't look like it's changed, but it is different. The atoms, the electrons, the positrons, all the trons have moved. And while it looks the same, it's not. In fact, everything, oops, oh, I've got the wrong button. Everything is probabilistic. And this is one of the most amazing things that we can realize, particularly if we apply it to ourselves. We're all accidents. And by that, I don't mean that, you're some sort of unplanned, pregnancy or something like that. What I mean is that we're statistically improbable. And this improbability is realized in every aspect of our existence. So, in fact, the only thing that I can talk to you about that's not improbable is that I can guarantee that within 100 years of this call, everyone here will be dead. That's straightforward. 117 of us in every 100,000 people will die each year of cancer. 102 in every 100,000 people will die of circulatory disease. 10 in 100,000 people will die of diabetes. 30 in 100,000 people will die of respiratory disorders. 33 will die of external causes, blunt force trauma, other self-harm and various other things. That's about 364 people in 100,000 every year die from those causes in New Zealand. Death and taxes, it's all unavoidable. But again, how and when you die is probabilistic. But at the same time, it's undeniable that our actual existence is linear. We're born, we age, we die. And that's an artifact of the universe that we're in, perhaps. Maybe in other realities, that's not quite the case. But in this one, it definitely is. And that, unfortunately, influences the way we think about ourselves. And, in fact, the DNA that encodes us and our traits. And indeed, many ways, that is really unhelpful. And as I go through my talk tonight, I hope you'll see why that is. So, to the question of my talk, how do we tap the untapped potential of our DNA to personalize your healthcare and extend your life? Now, many of you, I think, might not like this answer, but the answer is actually relatively simple. And that is that we need to change the way we think about healthcare. What we need to do is we need to move from a system that tries to correct or cure a disease to a system that is truly proactively trying to inform prevention using full patient participation. How do we do that? Well, in many ways it's very simple, and in others it's extremely complex. And I'm sorry to say, but for many of us on this call, it's a bit late to make a huge difference, because the diseases that are going to kill you have already started developing. You just can't see the symptoms yet. So what is the answer? Well, the answer is simple. The answer is that what we should do is we should sequence the genomes of all of our children. Not a representative few, not most, but all. It's not going to help you, but it is going to help them. And I'll explain why. Now, some of you will go, that's not possible. There are 60,000 children born a year in New Zealand. And I'm telling you, it is very, very possible. 24 machines running 24 hours, 7 days a week would do this. We'd get DNA sequence, we'd get epigenetic marks, which are the signals from the environment, and we'd get that all on those individuals in one run. 60,000 children a year born in New Zealand in Aotearoa. The materials to sequence, just the materials to sequence them, would cost $60 million. $60 million, that's all. By comparison, the Human Genome Project took 13 years and cost $3 billion US to do just one individual. But we've moved on. Things are faster. We can do this. In fact, if we really wanted to and we thought that $60 million was too much money, then we could actually do it cheaper. We could do it for about 6 to 9 million, but we'd only get about 1/10 of the information, or maybe even 100th of the information that was there. But it would be enough, definitely enough to start, definitely enough to make a big difference. Now listen, I'm not saying this is simple. There'll be hiccups. They'll come along the way, but eventually, over the course of a couple of years, it would become business as usual, and it would impact lives in ways that we have very little understanding now. Positive, very positive impacts. Now, before you say, we can't do that, it's not ethical, we already run very exhaustive tests on children using the Guthrie test, for example. This looks at metabolites in their blood, and it's done on all children, and it has helped save thousands of kids. Doing the DNA in the way I'm talking about now will address both common diseases, the complex ones like diabetes, cancer, Parkinson's, Alzheimer's, all of those types of diseases, and it will also address the rare diseases that are present in our population. And this is really important because there are 7,000 rare diseases. Well, who cares? 7,000 rare diseases. They're rare because they occur in 1 in 100,000 people or something like that, or maybe 1 in 10,000 people. So it doesn't really matter. Well, it does matter if you're one of those people that has it. But there's so many of these rare diseases, 7,000, that in fact, a rare disease occurs in 1 in 10 people, 1 in 10 New Zealanders. So currently there are 400 to 500,000 New Zealanders who are here now walking around who have rare diseases, many of which are undiagnosed, some of which can be treated, but they don't know. This is a huge burden on the people. Not only them, but their families, their whānau. And unfortunately, most rare diseases impact children. 50% of them impact children, and those ones, a lot of them kill the children before they're 5 years of age. Huge impact. So, now that I've told you what I think we should do, let me explain why I think we should do it. Let's talk about the DNA itself. Well, your DNA contains instructions that make you you. From a simple perspective, the information is stored in about 3 billion bases. These bases are adenines, cytosines, guanines, and thymines. They're arranged in 22 autosomal chromosome pairs and 2 sex chromosomes. And you inherit those from your parents, 1 chromosome each from each of your parents. The chromosomes range in length from about 250 million bases down to about 50 odd million bases. And they all contain different numbers of genes and various other aspects. You know, if you were to take the DNA out of an individual and extract it all and line it up end to end, it would go from Earth to the sun and back about 133 times. Just in one individual, there's that much DNA. And then, of course, there's actually a hitchhiker, a bacterium, which is actually part of our cells, and this is the mitochondria. And that has its own little genome and its own unique genetic code. And that's quite important to remember. But genetic variation in our genomes is quite diverse. They are very diverse. As Wayne said, there are about 44 million single nucleotide changes that we know that occur in at least 1 in 20 people's genomes. Between any two of us on this call, we have at least 1 million differences in our DNA sequence. There's 1 million differences between any two of us. Now, unfortunately, this information is a little bit biased because when I say the human genome, what I'm really saying is I'm meaning the Caucasian or the European genome, because the sequencing of genomes has been expensive up until now, and the work has been funded in places where it's led to an unconscious bias in the genomes that have been sequenced. But that has been remedied now as we've become aware of that bias. And in fact, what's happening now is there are projects like the Variome project, which is being run by Genomics Aotearoa, which is actually looking at genomes in diverse populations. And the Variome project for Genomics Aotearoa, which is an amazing project, is looking at, of course, the Maori and the Pacifica genomes. Now, most of those 44 million changes that I talked about actually occur outside of genes, because genes only make up about 2% of the DNA that you have in your cells. And that's a really interesting thing, but it poses quite a significant problem. It poses a problem because whilst the gene products don't guarantee anything, I mean, they're basically machines that increase the probability of a particular reaction occurring in your cells, we can see how things catalyzing reactions in a cell can actually lead to a trait, right? Because if you don't make a product, then you don't create a trait, or it doesn't help build that trait that you're looking at. It's like if you break a brick in a wall, then the wall can fall over, right? A very simple thing. But it does leave the question, how do these variants, these changes in our DNA that are outside of genes, how do they actually impact anything? Because they're not affecting a gene. And we've all been taught, since we've understood Mendel's rules, you know, genes are the units of inheritance. They're really important. Well, not that simple. And so that's what my group and many other people work on. And really, it's a major endeavor now, trying to assign function to these non-coding, these genetic variants that occur elsewhere in the genome. But we do know a few things about them, because sometimes you don't have to know how something works in order to be able to use it, right? That's a real simple thing, okay? We don't necessarily know how a television screen works, but we can use a television. Same thing with your DNA. And let me explain that. So genome-wide association studies are studies where we take large groups of people, we categorize them according to some particular trait. So let's say a simple thing is like hair color. And we'd take one group of people with blonde hair, group of people with brunettes, and then we would sequence those people. And we look for the changes in their DNA that actually correlate with the presence of the blonde hair or the brown hair. And by doing that, we can understand which parts of the DNA actually are associated with that particular trait that we're looking at. Now, hair color, it's not so complicated. Well, it is in many ways. But what we like to do, and scientists do, is they try to do this to understand the risk of developing diseases and complex diseases. So diseases like diabetes and Parkinson's and asthma, chronic obstructive pulmonary disorder, even things like COVID infections. And I'll come to that in a few minutes. Now, the amazing thing about this is that you can actually use this information. And so what I've got here that I'm showing you on this slide is some pictures of some people, okay? Now, there's a company, and there are a couple of companies in the US that have actually taken photographs of individuals, and they've taken 300 or more measures on the face, right? Simple. You take nice pictures where you rotate the camera around, you can get all these pictures and measurements on people's faces. What they've also done is they've sequenced people. And then they've done exactly what I explained, but instead of doing it for hair color, they've done it for their facial features. Interesting question, right? Can you do it? The answer is you can. If you do that, you can actually correlate facial features with features in the DNA sequence itself. And that's a really powerful thing, because the images that I've got on the screen here at the moment are computer reconstructions from people's DNA, all right? And this is pretty important. So this sort of technology is being used now in some cases where people are looking for offenders, or police or other people are looking for offenders who have done violent crimes. The question is it any good? Well, there are the images at the moment that you can see, which are the computer reconstructions. This is what this lady looks like originally, and this is what this, well, not originally, it's what she looks like in real life. And this is what this guy looks like in real life. Now, these, of course, are some of the better images that they have, but you can see how identifiable those people are. Now, we don't know what the genetic traits or the genetic variants are doing, or how they're actually correlated with this, but we do know that they do correlate with these features. And because we know that, we can use it to predict what these people look like. Now, you can do exactly the same thing for diseases. You don't have to know how it contributes to the disease, but if you know that it's associated with the disease, then you can predict whether someone develops a disease or doesn't. So it's really important that we don't just think about the usual suspects here, right? Candidate studies or candidate gene approaches have done amazing things. You all know of genes like cystic fibrosis, CFTR, or Huntington's. Candidate genes, very, very, very important. Absolutely. But complex diseases don't just involve those candidate genes. They involve many variants across a genome that work together, each one having a small effect, but contributing to the overall phenotype. And that is really important to understand. So we're not looking just for the usual characteristics here or features. Now, I know that you're probably sick to death of talking about it, but we just need to talk about SARS-CoV-2 for a moment, so COVID. Now, my daughter, and I call her 18837 now because that was her COVID infection number, you know, because we worked it out when she got infected based on the number of infections that New Zealand had had. So 18837, she got COVID. She hung out with us for a few days beforehand. It was a holiday weekend. She started to show symptoms. She got tested, went into isolation. We all did because being 18837 was relatively early on. But the rest of the family, nothing. And you've all got stories like that. We all know people that have been like that, right? They've been involved, very close contact for a long period of time with people, and they haven't got COVID. We all recognize as well that some people show symptoms and others don't. Some people will get hospitalized, others won't. Some people will die. Now, there's no real rhyme or reason to this as far as we can tell just by looking at people. And COVID, as we call it, of course, is a result of a viral infection. It's not what you would think of complex diseases. But I think that's where we're wrong. And that's one of the instances where the sort of information that's hidden in our DNA can actually be quite helpful in understanding why some people do and some people don't die from COVID. Now, there have been big studies done internationally because obviously we've had this technology here and people have used it. And they've used it to understand what's happening during this pandemic in a way like we've never been able to do anything before in any other pandemics, right? And it's amazing. The information is incredible. So there are 3,801 SNPs, or single nucleotide polymorphisms, or changes in the DNA sequence that have been associated with severe responses to SARS-CoV-2 infection. 3,801. If you happen to have the right combination of those, then you are going to be severely affected by SARS-CoV-2. You're going to wind up in hospital on a respirator and or die. Very, very high probability that that's going to happen. There are another 4,734, variants, which have been associated with being hospitalized. So these ones increase your risk of just being hospitalized. You don't necessarily wind up on a respirator and or die, but you will most likely wind up in hospital as a result of the infection. Now, naturally, if this GWAS approach works, right, and this information in the DNA is real and it does correlate with the severity of the infection, then there should be overlap between the ones that you wind up in hospital or the ones where you wind up in hospital on a respirator and or die. And yes, there is. So there are about 2,348 of them that are common to both. Now, these 2,348, they form switches. They form switches in the DNA that control how genes are turned on and off in the lungs, in the brain, in the heart, in the other tissues of the people who suffer from this infection. Now, that is the key to understanding why these people wind up, in part at least, why they wind up in hospital or on a respirator. But it's not those ones that are common. They're important. Don't take it the wrong way. But there are unique ones, and the unique ones are really the key to this. So there are 649 switches that affect lungs in people who are hospitalized, just hospitalized. There are 248 different switches that affect the lungs in people who are severely affected, who wind up on a respirator. Now, some of the people in my group have worked out a new analysis that you can do to basically understand what these switches are doing and relate them to the morbidities that are associated with your outcome for SARS-CoV-2 infection. So this is an analysis that we've done on other things, but it hasn't been published yet. So please do take this with a grain of salt until my peers have basically said, yes, this is a good thing or a bad thing. But what we think it shows is this. The switches that are unique to either being hospitalized or being severely and put on a respirator are impacting on biological processes and pathways in the lung. And they're doing that in a way that is really quite intriguing. So many of these variants, in particular the ones for the hospitalized people, there are a set of variants that impact on general lung function. Now that makes good sense, right? We can all see that, right? If your lung function is not so good and you get a lung infection, then you're going to wind up in hospital. That seems reasonable. The severe ones, well, those ones actually impact on lung function too, but they do it a different way. What they do is they actually impact on lung function in a way that is reminiscent or predisposes you to chronic obstructive pulmonary disorder and emphysema. So this is a really severe impact on your lung function. And so the genetics and the probability is, as a result of all of this being probabilistic, is that if you have that set of variants, your lung function is really severely impacted, just in general, predisposing you not only to SARS-CoV-2, but COPD and emphysema. Therefore, if you get that infection, you have a very strong response and you need respiration and respiratory help. Now, the people who are severely affected, they also have changes in things around interleukin levels and fibrinogen levels. And of course, fibrinogen is one of those things where you get lots of clots. It makes clots in the blood, and those levels are changed. So they're clotting changes. Again, these are things that we know from looking and using epidemiological studies that just look at the characteristics of people in hospital. But they also show things about getting rid of chronic hepatitis infections. And that's an antiviral response, right? So their antiviral response is not as good. Again, makes great sense. Now, we can understand how those traits might interact to make a person more or less susceptible to SARS-CoV-2, right? We can all do that. That doesn't take a rocket scientist to do that at all. But some of the features that come up are not so easy to understand, and yet they are very statistically and biologically very robust calls. And they're very interesting. One of them is an interaction with Parkinson's disease. Now, just to be really clear here, I'm not saying that everyone who gets COVID is going to get Parkinson's disease. But we do know that after the Spanish flu epidemic in 1918, there was a significant increase in the rate of Parkinson's disease that was diagnosed. There seems to be an interaction between these variants that predispose you to being affected by SARS-CoV-2 and Parkinson's disease. And the interesting thing about this is it's something that we've seen in the epidemiology, at least of the Spanish flu virus. A similar, well, not similar, but it's a different respiratory virus. So our concern is that that's going to happen now. But we also see interactions or evidence for interactions with other chronic conditions. Coronary artery disease, again, a well-known morbidity or comorbidity of SARS-CoV-2 infection. Multiple sclerosis, mood disorders, type 1 diabetes. These comorbidities have been identified in these massive epidemiological studies of populations who are affected by SARS-CoV-2 or COVID. But the power of the analysis that we've done using the DNA and starting at the DNA level is that it doesn't just say these are the multimorbidities. It identifies those multimorbidities without having any a priori assumptions of what they might be. We made no assumptions about what the multimorbidities might be. We did it in a completely uninformed way. And when we did that, we found not only the multimorbidities that have been seen in these epidemiological studies, but we also found the genetic variants that predispose you to that. So therefore, we can calculate a person's risk. And we also found the biological connections that are actually responsible for that. Now, that means that not only do we know the trait to look at in the individual, but we know the genetics that give them the risk, and we know the biology that's behind that, which means that we can target in ways to modify, potentially, those multimorbidities and maybe affect the outcomes for these individuals. That's the power that sequencing the DNA of individuals gives you. It gives you that ability to where you have an understanding of the things that might work together to contribute to a disease like diabetes, Parkinson's, any chronic disorder like that, that you can actually work out what might be contributing to it, and you then know how you can treat an individual, okay? And that's really the power. So, unfortunately, for COVID, it is a case that even though they say a mask and gloves are enough to go to the grocery store, they do lie. Everybody else has got their genes on, all right? Their genes or their SNPs, probably more importantly. Okay, yeah, bad science pun. My son will tell me off later. But we shouldn't think about diseases in terms of just one particular condition, and that's what that tells us, right? SARS and the COVID infection. COVID is a complex disorder. It's a result of multiple things happening, the infection interacting with other morbidities, and your outcomes depend on those morbidities. And so rather than thinking about things as just one index condition, we should think about things as being a set of conditions that work together. All diseases are like that. There's not one form of multiple sclerosis. Your form of multiple sclerosis is unique to you. It's unique to your genetic risk. It's unique to your environmental exposures. Your form of Parkinson's is unique to you. It's your genetic risk, your environmental exposures. The same for any chronic condition that we suffer from. Okay, so your disease is unique to you. And if that's the case, then we should treat each disease as if it's unique to people, because giving people the same drugs doesn't necessarily mean it's going to have an impact on everybody in the same way. And we know that because, unfortunately, in New Zealand, well, fortunately in New Zealand, your lifespan is into the early mid-80s. If you're European, if you're Maori Pacifica, it's sort of 10 years shorter than that, which is an issue that needs to be addressed. But roughly from your 50s, most of us, or 50% of us at least, will have at least one condition that we get treated for on a regular basis. One condition. As you age, you will end up with most likely more than 8 conditions that you're being treated for at any one moment in time. Now, those conditions aren't independent of each other. They all develop together and they interact together. And many of you who have had older loved ones and they've been treated for these conditions will know that when they get given one drug, sometimes you've got to give them another drug to balance out some other effect on another condition that they have to try and bring the things together, because the drugs cross-react with different conditions. That's because the genetic and the biological basis of those diseases is not independent. They are intertwined, they work together. And that's what we have to do. We have to understand that, how they work together. We can do that if we sequence the genomes. And that's a really important thing, all right, because over the course of your lifetime, you're going to have about 14,000 pills as you go from cradle to grave, all right? 14,000, it's been counted. And that's the sort of average number that somebody in the UK has. But 14,000 pills, it's mind-staggering when you think about it, right? Just how many pills that is. But you could change that if you reduce the frequency of these conditions. Reduce the frequency of these conditions. In order to do that, you want to do it in a progressive way. You want to empower people to change their lifestyles, to not do the things that are going to put them or exacerbate their genetic risk. You do that by telling them what their risks are. So, how would sequencing our children help? Well, it's simple. Many traits now, we can estimate the risk for a disease. And we do that by combining those changes across the genome, pulling them together and saying, these ones all contribute to a risk of your risk of this disease in this way. And your overall score for that is high or low. So you have a high risk of developing it or a low risk of developing that condition. And in some instances, not in all instances, I'm not saying in all at all. I mean, it's got to come and there's more that has to develop. But in some instances, our understanding that risk is good enough to use it already in the right populations. So for example, type 1 diabetes. Your body stops making insulin, your pancreatic cells are destroyed, or your beta islets are destroyed by your autoimmune system, right? We kind of know how that works. There are about 70 genetic variants in your genome that will predict your risk of developing type 1 diabetes by the time you're 5. 70 variants by the time you're 5. If you know that a person has that risk, you can monitor those persons for the onset of, or you can treat them to try and alleviate and prevent the development of the autoantibodies that destroy their pancreas. And therefore, you will extend the period of their life without type 1 diabetes, which before the development of insulin was a death sentence, and a horrible death sentence, right? Drink lots of water, starve, die. Horrible. But type 1 diabetes is not actually just a disease of kids. 40% of individuals who develop type 1 diabetes will actually present after the age of 30. 40% of those people will have actually type 1 diabetes. There's a lot of misdiagnosis that occurs. And I'm sure Wayne will talk to me about that later. But polygenic risk scores, okay, and that's what we're talking about here, where we take multiple little snips and we add them all together. These scores tell us the risk of developing a condition across the life course, okay? So it doesn't matter when the disease develops. These things will tell you the risk of developing that disease across a person's life course. So from the moment they were conceived through to the moment they die. And that's because the genetic variants that make these scores up or that contribute to these scores are inherited from your parents. Now, there are catalogues of polygenic risk scores. We catalog everything, right? There's catalogues online. So there are about 2,199 polygenic risk scores for about 538 different traits. So think about that for a second. 538 different traits. Many of them are diseases. If you sequence the genome, you can look at those scores. Now, right now, a lot of them aren't going to be very accurate, but the more we do it, the more accurate they get, the better it is. It's chicken and egg sort of scenario. So whilst the sequencing would be done on children, we're predicting what's happening to them over their life course. Effectively, we can tell them you're at risk of this disease when you are 40 or 50. Do these things now, monitor these things, and it will push that disease back. It extends their well-being and their life. Really important. And that's where this comes in. That's where the power lies. Of course, the scores are only as good as the data we've got, right? So we've got to build more data. We have to do this. And we're going to do that by iterating. We're going to do that through projects like the Varian project that's been run by Genomics Aotearoa, where all of those things are really important. You know, but we should have learned from Google and the electronic exhausts that we have, right? Because what I'm talking about is not complicated to do. As I said, 24 machines or 48 machines, 24/7, we will be able to sequence everybody in New Zealand. $60 million. Everybody born every year. That's not that much. Have we learned from Google? Do we really want companies like 23andMe, who we and many of our friends and relatives have sent their DNA to, do we want them to do this for us? Do we want them to own this information about us? I don't think so. Maybe you disagree. We'll find out. So risk scores, they do work. People who are in the top 8% for coronary artery disease, polygenic risk scores, have a risk that's comparable of developing coronary artery disease that's comparable to really significant monogenic disorders. Okay, familiar with hypercholesteremia. But if you tell those people who have that polygenic risk that they have it, and this has been done, knowing it empowers them to change their risk reducing behavior, or to change their behaviors, and actually gives them more personal control, and it actually reduces their overall risk. As I said, we still don't know what the genetic changes are actually doing and how they're conveying that risk onto these people, but we do know that it works, and that's important. So, the benefits of sequencing our children, or in fact, ourselves. You might be able to squeeze a few extra years out of it, you know. Patient stratification, that's simple. If we know the complex diseases that are working together, how they interact to cause a disease or link to one another, then we can actually stratify patients. We can do it and we can use that information in clinical trials to actually understand why some of the people in clinical trials respond and others don't. That happens in all clinical trials, but if we don't get enough responders, because the clinical trials across a cohort of people, we say it didn't work. But that's not right. The people are individuals. They have their own risks. Those risks are what we should be testing against. Some of those drugs actually do work. There's a recent Alzheimer's one, which is a classic case in point. The stratification will also help with your best treatment strategy. If you go to a doctor and I've got this risk, here it is, these are the things that are affected, they know that there are drugs already that they can repurpose and target those things and might in fact help with the condition that you've got. Early diagnosis for people with rare disorders, and don't make a mistake, this would make a massive, massive difference to these people. High-tech industry will develop as a result of this. Our health research would be able to be focused to treat our own population. We're looking for ways to impact our population. The government gives us the money to do the research. Why don't we do it in a way that impacts our own populations in a way, that makes a really huge impact? We could, and we have the skills in our country, we could be targeting individuals and working on them as opposed to maybe some of the ideas that I might think are interesting but aren't really going to help anybody in the short term. Equity in treatment, reducing false hope for treatment with drugs that we know are going to have a slim chance of working in individuals, some individuals, and empowering people with the knowledge to help themselves. But I'd be lying if I said they're just positives. there are a lot of issues associated with this, okay? Firstly, we don't yet have the capacity, the workforce to do this. But there are projects underway now that will change that here in New Zealand. We have issues with consent. We have issues with social license. Guardianship of the information is really important. How do we guard our own information and information about this? Insurance. I don't think insurance is really a problem with this because we have a public healthcare system. And that public healthcare system, if we take pressure off it at the top end by just trying to remedy people and patch them up, and we actually start preventing people, it's going to give more money to the system effectively by shifting things around. We don't know what the variants do, but we will learn from them. So that's pretty much what I've got to say. But I do have one last thing, and that is that like all scientists, we think that we discover things anew. But precision medicine and precision approaches using information which comes from your DNA have been used for a long time, okay? You've all been asked about your family histories. You tell the doctors this, it helps in your diagnosis. You've been done blood typing, again, DNA-based. Transplantations, tissue typing, DNA-based. Clinical scores, all on the basis of features that you have, characteristics that you have, are not doing it using your DNA sequence, but the characteristics and the features are a result of that genetic risk you have and in environmental exposures that you've had over your lifetime. So we might not always recognize it, but DNA lies behind many of the things that are already happening to us in ways that we're treated clinically. So just to finish, this is a difficult conversation because it's challenging the status quo. But I hope that the conversation we've had has challenged many of you to think about this. And I personally believe that we must democratize this for our own children's sake. Now, this type of work is really difficult to fund, but we're really lucky that we've recently received quite a significant donation from a family that want to see this type of work undertaken in critically ill children. And I'd like to thank them for that, and I'd like to thank you all for listening. And Wayne, hit me.
Kia ora tēnā koe, Justin. That was very informative and certainly provocative. We have some questions, and I'm going to slightly alter the order of the questions. I'm going to start with a question, Justin. And the question is regarding the SNPs that make you more likely to get seriously sick, like with, oh no, it's not that one. Okay, if you have a genetic variant that results in you developing a condition, oh, no, sorry, okay. No, let me back up, okay. If you have a genetic risk that you can't do anything about, what do you do about that? And should you inform the person?
Oh, yes.
I guess it brings up this whole ethical dilemma that exists in looking at the role of genetics in diagnosis. Now, what you commented on later on is kind of the new frontier of medicine, which is, and I see this as a clinician every day, and that is we don't treat people with the same condition the same way anymore. We look at what their risks are, and some of those risks will be genetic risks. And as you've said, cancer is a huge example, where if you have breast cancer, you don't receive every patient doesn't receive the same drugs as they did 10 years ago.
That's right. Yeah.
It depends upon the genes that increase the severity of the disease.
That's right.
The responsiveness to treatment. But where it gets a little more challenging is where you are using the genetics earlier on to look at who might or might not get the disease. And I'll come back to your comment when you started, which was the most provocative one of all, which is sequencing the genome of all of our children. You know, the Huntington's dilemma is a big one. Do you screen or don't you screen? And for each family, it's a problem that they face because there's no cure. There's no cure. Yeah.
So I think actually that, I mean, it's a very good question, right? And I didn't cover that very well. Obviously, you know, some fields have been moving a lot further than this in this area than others, and cancer is one of them. And you know, guys like Chris Print, who are here at Auckland University, are doing amazing work looking at, you know, using DNA and the way they treat people and do these things, and others as well. It's just the name that pops in my head right now. But the question about what do you do with these unintended ones, the ones that you can't treat. Now, there's two things. A, you cannot report, and there are guidelines which come out from the clinical genetics, American geneticists, which are these ones you should report on, these ones you shouldn't report on if you identify them. But you come back to some things, and I've asked people, about ApoE4. So ApoE4 is associated with a very high risk of developing Alzheimer's if you have that particular haplotype. I've asked people. Now, some people say they don't want to know because you can't do anything about it yet. I think those treatments are going to come. They're going to come for that. They're going to come for Huntington's. They're going to come for other things as well, right? Because you can actually use CRISPR to modify the DNA, right? And clinical trials like that are happening. In fact, there's a new one coming soon into New Zealand, which is going to look at cardiac. But those types of trials are happening. But the important thing is that some people say, I don't want to know if I've got ApoE4. They don't want to know because you can't do anything about it. And yet other people will say, I've got ApoE4, I want to know because I'm going to change the way I live my life. And I think that's a different thing, right? Because you're empowering people to make those decisions. Even asking them, do you want to know, empowers them. And so I think that people have a right to ask that question or to know if they want to. And if they don't want to, then that's fine. But I think a lot of these things, and the treatments are going to come. I mean, the technology is moving so quickly around CRISPR, around the way that we can modify DNA and change things, that these things are not going to be, I think, untreatable in maybe not in our lifetimes, but maybe in the next lifetimes after. I think treatments are going to be there that are really good.
Okay. Now, there's a recurring question here, Justin, and that is when somebody's told they have a genetic risk, there's a risk that they might just kind of give up. In other words, they could potentially manage that condition by making an effort with lifestyle, but they don't because they've got the genetic condition. Why bother?
Yeah, so that's completely about our understanding of risk. And that's a good question, but we have this real problem with risk. I think casinos, one-armed bandits, those sorts of things tell us that as a group of people, we have a problem with understanding risk, right, and probabilities. It's one of the things that we have to work on. Just because you have a risk doesn't mean you'll develop the disease. That's it. It's a genetic risk. There are some things where you have a very high probability, but even with cystic fibrosis, you can have a mutation that should give you childhood onset of cystic fibrosis. And yet there are people walking around with that mutation, 2 copies of it, so they should have childhood onset, and they don't have any symptoms. And they're adults. Now, that means that there are other things that are modifying the way that is realized, the way that gene works in those people, and it's stopping them developing cystic fibrosis. The more we understand about the way these genes work and the way these variants work by looking at bigger populations, the more exceptions like that we're going to find, the more novel ways we're going to have to actually treat people who do show the symptoms early, where the risk is being realized.
Isn't it fair to also say, Justin, is that our outcomes are dependent upon our genes, but also our lifestyle and our behavior and well-being?
Oh, yeah, absolutely.
And that you can moderate risks appreciably.
Yep.
If you radically change your lifestyle.
Absolutely.
And so that is the one thing that that's the one variable we can control. We can't control our genes, they were set for us, but we can certainly modify our lifestyle and behavior. Yep. And that potentially can modify genes genetically. But
you're right. Your DNA is set when you get it from your parents. Your risk is set at that point. And so it's the interaction between that risk and your environment. That's what ultimately determines it. The environmental signals you're exposed to, undoubtedly. Drink too much, smoke too much, dunk yourself in chlorine, really concentrated all the time, I don't know. Just chemical exposures, all those sorts of things will modify that risk. And some of them will trigger it off and you will develop a disease as a result. But I think that is definitely modifiable, your environmental exposures, right? You can modify your behaviors, you can modify what you're exposed to. Those things you can change, you can control. But I think the big difference that's coming now, as I said about this CRISPR, is CRISPR is basically molecular scissors that cut your DNA and replace a bit of it, right? Soon, we'll be able to actually do that, I think, in a way that actually allows you to modify not just your environmental exposures, but also your actual genetic risk as well.
Now, if you have a genetic variant that results in developing a condition, can this same genetic variant trigger other conditions? In other words, one genetic variant seems to lead to a snowball of different conditions.
Yeah, so we know that there are certain parts of the genome where you can get different variants, but they're all close together. Those regions actually do that, and we call them like super enhancers. You know, they control many genes, and that impacts on many traits at once. So those types of regions are regions that we look for a lot, but also many of the genes are controlled by many different parts of the genome. And so you can have things that are associated with COPD, for example, that are at one part of the genome, and Parkinson's, and they interact together, but through a gene that they both control. And so there are different ways of this, but the answer is yes to the question. Yeah.
Just coming back to COVID again, the SNPs that make you more likely to get seriously sick with COVID, do they just affect respiration only? And I'm, projecting on from the question, of course, it brings up the big issue, which is sort of chronic COVID, brain fog, and in the small portion of patients who it seems to affect brain size even, is that likely to be a reflection of that person and their genes and the virus, and not just the virus itself?
Yeah. So Rachel's been doing this work, and she's shown that, in fact, it is different in different tissues. So, the variants do different things in different tissues, and that's because the way your DNA is organized, information is not just in a linear string of DNA, but it's in the three-dimensional organization of that DNA. It changes between tissues. And so you actually see that in different tissues, you can see some of the morbidities that actually seem to appear and be linked to COVID infection as well. So we think that's very tissue specific. And that comes back to this whole idea that really we should be looking at the person as a whole system and not just as a series of organs or a series of cells or something, right? But that's a really difficult thing to do. It's to put it all back together in one big piece. Yeah.
Now, here's a really interesting question. In the world we live in right now, where there is grave concern about data on us and how it is used, and we're all aware of the Facebook saga and the way in which Google tracks and monitors us.
Yeah.
How do we protect our genetic data, and who protects it for us? Because if it was done on any scale, or even if I decided that I wanted to have some genetic testing before, where does that get held? Who protects it? Who protects it from big pharma? Who protects it from life insurance companies?
Yeah, okay, so it's a good question, all right? But do you actually have to keep it? So if it costs $1,000 now to do a genome, right, then maybe in 5 years' time, it's only going to be $100. All we have to do is, do we actually have to keep it? Do we use it? Do we sequence? Do we get the information out of it?
Yeah.
These are your risks. And then
But how do we know that it hasn't been kept? We might want it deleted, but how do we know that it's?
Well, I think I don't.
Somewhere.
I'd love to say I have all the answers, but there are people that are way more qualified that think about these things in way different ways than me, and much more qualified to talk about sort of the ethics and things behind this, and the way you store the data and the way you look after it and the guardianship. I mean, and there's been a lot of work that's being done on that, and so I think, like I say, I'm not really the best person to ask, but yeah, sorry.
I mean, it's a really good question.
It is a good question.
Given the issues around data management, and of course for Māori data sovereignty is an enormous issue right now, with all of the government data sets that exist. What you commented right early on, before we finish, I'll bring you back to it, was to sequence the genome of all of our children. And so the question was, when's the best time to do it, at birth or later on? If you're going to sequence everyone, when are you going to do it?
Honestly, I think you may as well do it straight after birth. There are many conditions that will pop up very shortly after birth. If you do it early, you do it quick, you might be able to find those. And there are things that you can actually treat. You can stop long-term damage if you can find them and you can treat them. Why wouldn't you do it early? Why would you wait and let some of those things impact on the children that we're trying to help? My thoughts.
Now, at what age do you talk to an individual about their genetic risks? And of course, in part, that might depend upon whether the disease is actually treatable now.
Yeah, so I think, I mean, that's another very good question. You know, how do you implement this? I mean, obviously, you wouldn't be telling a baby that this is your genetic risk, because the baby just wouldn't understand, right? So you've got to tell the parents. And I think the parents have got to inform the child and do that through the way that obviously they're brought up. You know, that's clearly something that you would have to do. So that would be through families that you'd have to do that. I think that's the only way I can think of doing that, and maybe that's wrong. Maybe there's a better way, but I'm not aware of that.
I think from my perspective, Justin, I don't think there is a right way or a wrong way. I think it's a you know, this is one
good question.
This is where I think, you know, all of the issues that science is galloping ahead of how we manage the ethical, social, emotional, financial dilemmas that we're facing here.
Oh, without a doubt. Yeah. The technology is going forward at a rate that's way beyond the speed at which we are preparing ourselves for what it can actually do, and for the positive impacts. And indeed, there are negative impacts, don't get me wrong, but the impacts that it can actually have on us as individuals and as populations.
Here is the penultimate curly question, Justin. Given that we've already got ethnic disparities in diseases and outcomes, do you think genotyping could accentuate those disparities if we find more disease-related risks in certain populations? That could be Maori, could be Pacifica, it might be other groups. Could that?
Yeah, so I think as first point, I think ethnicity is a social construct. You're talking about ancestry. And so ancestral groups, they do have gone through bottlenecks. There are changes in their DNA. I think the beauty of the way that we're starting to look at this is that we're looking at the total regulatory or the total suite of the regulatory switches that occur across the whole genome. Now that means that we're not looking at individual ones that maybe have an impact in one ancestral group. We're looking at the ones that impact on all ancestral groups, which means I think that we're trying to move beyond the sort of ancestral bottleneck issues that we're talking about here. I think that this can be applied equitably across everybody. And I think that technology, as these technologies develop forward and the treatments go forward, we need to do this across everybody. And it has to be done on everyone, not just some people, some subsets of the population. This is a technology, and it's something that promises to redefine the way that we treat ourselves, that we look after our own health. It's important to do it to everybody. Personally, I don't think that any population is more disease prone than any other. I think we all have genetic risk in our DNA. So this genetic risk is there for everybody. And it's very important to consider it that way, I think.
Justin, thank you. You were informative. You were certainly entertaining and absolutely provocative, because from the Q&A, and we have no more time for Q&A. There's lots and lots and lots of Q&A and some great questions. So thank you very much. A wonderful evening. Just in wrapping up, to remind everybody, as Jody mentioned, the last Raising the Bar presentation next week is Bodo Lang, whose topic is I Told You So, Word of Mouth, What Is It? Does it work? And how can I use it? Otherwise, kia whai, ahi ahi nui koi. Have a great evening, everyone.